Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have been discussing mapping process, and in the last class we have introduced the Bowtie2 tool, right. So, this is a tool that utilizes Biomedical transform with FM index and LF mapping to map reads. So, what we have discussed so far is that how do we actually utilize this tool, how do you set this tool up, and how do you actually build the index, right. So, in this class what we will be doing is we will be actually doing the read mapping using the index that we have built up in the last class. Okay. So, just to remind you, right, we have set up Bowtie2, right. In the last class we have downloaded and we have extracted the tool, and it is a very simple process. It does not require very complex installation. You download, extract and check for permission, right. If it is required, you can change the permission using the chmod command, right. So, once this is done, we can do the genome indexing, right, using this function called Bowtie2 build, and we actually utilize that function in the last class, right, to build the index for Saccharomyces cerevisiae genome, right. So, we downloaded the reference genome from NCBI RefSec for Saccharomyces cerevisiae, because we are dealing with data from this organism. Okay. And during this build function, what is happening is that Biomedical transform is happening, then followed by creation of this FM index, double indexing and checkpoints, right. So, you actually have seen, right, this double indexing part, because we have this forward index. Of course, there is no name for forward index, these are like 1, 2, 3, 4, and then you also have the reverse indexes, right. We have seen those. And also we talked about the options. When you are running Bowtie2 build, you have certain options that you can utilize if required, right. If you want to speed up the process, if you are building this index for a really big genome, like human genome, you probably want to speed up, you want to give this minus threads option, and if your system permits, right, if you are working on a server, you can utilize this minus threads option to speed up the process. Okay, so, we have discussed up to this point. What we are going to do today is we are going to actually do the read mapping with Bowtie2. Okay, so, there is this function Bowtie2 that will do the read mapping. So, in that way it is a very simple process, but of course, there are few other things that we need to know about when we actually do the mapping, right. Those parameters could be important during the mapping process. Okay, so, let us first look at the command, how we will actually run this to map reads against the reference genome, right. So, this is how we will specify the command. Okay. So, the command is Bowtie2, right, there is no build or anything, just simply Bowtie2. Then you have some sort of options, and we will come to these options later. Unlike Bowtie2 build options, these options are very useful and in some cases critical. Okay. So, we may have to specify certain options to get better quality results. Then you have something called minus x BT2 IDX. Okay. So, what is this minus x BT2 IDX? It actually specifies the index file. Okay. You remember we gave the index file name when we are building the genome index, right. So, we mentioned east underscore WG, right. Here you would have to mention this minus x east underscore WG, that base file name. Okay. We do not have to mention east underscore WG dot 1 dot BT2 etc. We just simply give this east underscore WG, and it will automatically take, right, all the index files, okay, in the processing. Okay. So, the system knows, right, this program will, this command will know, right, okay, these are the BT2 files, it will search for them and it will take up those files for processing. Then you have some commands within these brackets. Okay. So, this is actually specifying the input files, okay, the FASTQ files, right. So, we are working with FASTQ files here. So, this will be FASTQ files. Okay. And you have three options. So, these three options are separated by these separated signs, right. So, you can have this minus 1 M1, minus 2 M2 options, or minus U option, or minus interleaved option. Okay. So, these characters that are between this less than and greater than sign means here you would have to specify certain value, right. This is where you should give your values, right. So, for example, here also that BT2 IDX you see this greater than less than sign, which means here you will give your file name. Okay. So, this is a way to actually specify where you would have to, the user would have to specify file names, etc. Okay. So, what do this mean actually? -1, -2 will come in a moment, and then you have something called -s and then SAM means this is the some sort of file that you have to specify again. Okay. So, as I have mentioned -x BT2 IDX, this is base name for index file for the reference genome. So, in our case the base name was east_wg. Okay. So, that will specify here. Then you have something called -1, -2. So, -1 M1, -2 M2, these actually these -1, -2 they are used together, and they are used only when we have paired n data. Okay. So, if you are working with paired n data, you will have to specify -1 the first file name, first Q file, and then followed by -2 and the second first Q file. Okay. So, you will have let us say -1 east read 1 dot first Q followed by -2 east read 2 dot first Q. Okay. So, this is how we specify paired n data. Okay. So, this is the way the program will know okay, we are dealing with paired n data, right, and try to align them in that way, right, for paired n data specifically. If you are working with unpaired data, you will just simply mention -u, right, and the file name. Okay. So, -u east dot first Q, right. So, that will be the format for if you are working with unpaired data, that would be the input file name. Okay. Now, there is an interesting format which is the -- interleave, right. So, this does not happen in Illumina, right. So, where you would have file that will contain read 1, read 2, interleave means one after another. So, you have let us say in the file you have read 1 sequence followed by read 2 sequence, then you have another read which is read 1, then the read 2 sequence for that corresponding read. Okay. So, this is the interleave format, but this is something that we will not deal with, right. We do not see this in Illumina, right. So, we do not have to use this option. Then you have the -s or SAM, this is actually the output file. Okay. This output file will contain the mapping results or the alignment results. Okay. And this file comes in SAM format .sam. Okay. So, you can say simply output.sam or east output.sam, something like that, right. So, that name should be given, so that the program will store all the results in that file. Okay. So, you might ask like what is this SAM format, etc. We will discuss in subsequent classes, right, why the data is stored in SAM format, what kind of information we get, that would be for later classes. Okay. So, once this is clear, right. So, you can now also can have this option -q means reads in M1, M2 or R dot first Q file. So, this is kind of a default, this is kind of understood, you do not actually have to mention this -q. Okay. So, what are the options here, right? This is actually important now. So, these options will come, right, and this can be classified into 3 different classes. So, you can have input options, you can have run options, you can have output options, right. So, for input options you are specifying to the program, right, how we should deal with the input. Okay. So, the input files that you have mentioned, how the program should deal with those inputs. So, that will be the input options. The run options would be the how the program should do the mapping, whether there are specific parameters it should keep in mind, right, what specific kind of choices it should keep in mind, right, that will come under run options. And then finally, you have output options, right. So, these are options that will specify, right, what should be written on the final SAM file. Okay. So, you can specify okay, do not write this or do not write that, etc., or do not map this, etc., right. So, those kind of options will be the output options. Okay. So, let us look at the input options first. Okay. So, here are the options. Okay. So, the options will be specified by this -5. Okay. So, you run Bowtie2, then say -5. Okay. So, you have seen this throughout now in Linux command line, we have a command, and if you want to specify certain options for that command, we use this minus something, right. So, that is the common notation. Here also is this is what we see. It is either -5, or you can also say -trim 5 and you have to specify an integer number, right. So, what does this -5 or minus trim 5 means is that you trim certain number of bases. So, this certain number will be specified by you, right. You can say trim 5 bases, right. So, this integer could be 5, trim 5 bases from 5 prime end of each read before you map. Okay. Why you do this? Because sometimes in your data you might see, okay, the first few bases are of very poor quality, right, maybe 5 10 or 4 etc. If you see that kind of during your quality control, right, you want to mention this, right, you want to do this, right, you want to say, okay, trim these 5 prime end, 5 bases in the 5 prime end and do not use them for alignment. Okay. The default value is 0, right, the program does not take any other, it does not do any trimming, it will just set this to 0, but if you want to do this, you have to mention, okay, -5 or minus trim 5 followed by the number of bases that you want to trim. Okay. And similarly, you have another option which is -3 or minus trim 3, okay, followed by this integer number, and it will do this trimming from the 3 prime end or the right side of the read, okay, before it does the alignment. Okay. So, again the default is 0. Then you also have other options, something called minus trim 2, 3 prime or 5 prime and followed by an integer number. Okay. So, what it does is that it will trim reads exceeding certain number of bases, okay, and you can also do something, right. You can so once you mention these number of bases, right. So, bases can be trimmed from either 3 prime end or 5 prime end, but they will not be done from both, right. So, you have to mention which side you want to do, right, whether 3 prime end or 5 prime end. By default, if you do not specify, Bowtie2 will trim from the 3 prime end on the right side. Okay. So, again this is a different option, right. So, if that reads that exceeds certain number of bases, you want to trim, right. Again, probably you know, like beyond, let us say 40 bases or 50 bases, there is no point in reading the data because the quality is very poor. In your if you are working with such a data set, you want to, let us say, trim beyond 50 bases, right. So, when you are running this Bowtie2, you can specify, okay, do not align beyond these 40 bases for mapping, right. So, this is something that you can also mention. Okay. And another point you need to know is that trim 2 and -3 -5 that we have just discussed, these are mutually exclusive. You cannot mention both, right. So, you cannot have -3 as well as minus trim 2, right. So, you cannot combine multiple options among these two, right. Otherwise, you can actually combine multiple options and we will see, right, how you actually do that. Okay. Then there are two other options, input options, which are also very useful, right. So, the earlier options, they were related to trimming reads, right, for mapping. While mapping the reads, the Bowtie2 will just trim the ends and then do the mapping. Here these two options are they relate to quality of the bases, right. So, when Bowtie2 is aligning, right, so it will read the bases along with the quality score, and the quality score can be in FRET 33 format or in FRET 64 format, right. So, we have talked about this FRET plus 33 encoding and FRET plus 64 encoding, right. So, older Illumina data, they utilized FRET plus 64 encoding. So, if you are working with old Illumina data, you want to mention this in the option, right. So, if you are working with new data, it will be FRET plus 33 and this is the default option now. Okay. So, there are other options now, which are the run options. We have talked about what are the run options, right. So, what we mean is that how you actually do the mapping or the alignment. Okay. So, these will come under run options. So, what you have seen now, the input options, they talk about the input, right, the quality of whether they are quality scored or the sequence streaming, right, they all talk about input. Here in the run options, it talks about the mapping process. Okay. If there is a specific way that it should do the mapping, or there are certain parameters that needs to be specified for the mapping process, those will be done here in these options. Okay. So, there are two useful commands that we can use. So, one is called end to end mode. Okay. So, this actually means the full read will be aligned against the reference sequence. Okay. So, you have perhaps learned about global alignment, right. So, where the two sequences, if you are aligning against each other, right, you look for full alignment, right, from one end to the other end. Okay. So, you do not leave out any part of the sequences. Okay. So, it is similar here, the idea is very similar. you align from one end of the reed to the other end. Okay. So, this is the end to end mode, is very similar to the global alignment. Inside this end to end mode, you can also say very fast, right, minus minus very fast, minus minus fast, or minus minus sensitive, or minus minus very sensitive. Okay. What it means actually. So, very fast means you want a very fast mapping, right, because you are dealing with a huge amount of data. So, you want to do this very quickly, but in the process you are willing to sacrifice certain accuracy. So, one of the things you should remember when you do this read mapping with BWT based methods such as Bowtie2, the backtracking process, right, or mapping reads with mismatches, right, that may not always give you the best results because of the certain features of the algorithms, right, certain features of the implementation. When you are mapping reads with mismatches, it might not give you the best results, right. So, unless you probably run this multiple times, you will not get the best results, right. You have to exhaustively search all the alignments, only then you might end up with the best result, right. But this exhaustive searching or exhaustive mapping is very time consuming, so you often ignore that, right. So, there might be some inaccuracy, right. So, the Bowtie2 may not always find the best match in case you have reads with mismatches. Okay. So, here what you are saying is that we can sacrifice certain accuracy, right. We do not really need absolutely the exact or the absolutely best match. We can sacrifice certain accuracy, but we want speed. Okay. So, you are sacrificing accuracy for speed. You can then you have these different levels, right, where you have this balance between speed and accuracy, right. So, if you come down to fast, minus minus fast, it means you want slightly more accurate, right, and you actually can sacrifice some speed. Okay. And then you have sensitive and very sensitive. So, very sensitive is the slowest one. So, it will actually do the exhaustive searching, will try to find the best match for all reads, right, and this will be the slowest, but it will give you the most accurate results, right. So, if you are working with small data set and you want very accurate ones, accurate results, then you probably run with this very sensitive option. The default option is minus minus sensitive, right. So, if you are running this in end to end mode, this is the default option, and this actually works quite well. So, as you will see, that once you have kind of run a lot of these or utilized a lot of read data for alignment, you will see this option is actually good enough for us. You can also run in local mode, which is actually in contrast to the end to end mode, right. So, local mode means you are doing something called local alignment, right. Again, this is very similar to local alignment, which means when you are aligning two sequences, local alignment only look for matches only in the certain parts, right. You will not try to do the end to end mapping, right. So, it might kind of trim or something called soft clipping here. It might leave out certain bases when it is aligning, right. So, it will not align the full read, and in some cases this is very useful, right. Why? Because as you have seen in Illumina especially, your quality score suffers at the ends, right. So, in the first few bases and last few bases, the quality score is low. So, if you are doing this end to end mode, sometimes what will happen, the Bowtie2 program, it will force this alignment against the reference sequence, right, and which might not be the best alignment, right, because of the sequencing errors, etc., in the ends of these reads. Okay. So, if you are running in local mode, what it will do, the aligner will do, is it will kind of soft clip those ends and it will find the best match, right. It will not try to forcibly match everything against the reference sequence. Okay. And so, in that case, it will probably find better matches in the reference sequence. Okay. So, in some cases, this minus minus local mode is very useful, and we use this option. Again, as in end to end mode, we have again different options. You can say very fast local, sensitive local, and very sensitive local, right. Again, there is this speed and accuracy trade off. So, very fast will be as the name suggests, right, it will be quick, but it will sacrifice some accuracy, whereas very sensitive local is at the other end of the spectrum, right. So, it is very slow compared, if you compare with very fast option. So, it is the slowest option of this all, but it will give you the most accurate results. Again, the default is this minus minus sensitive local, which actually works very well for most of our applications. Okay, so there are other options that we also use. These are called output options. Okay, so these are also useful in some cases. For example, if you are aligning paired end data, you want to specify what you will consider as a valid paired end alignment. Okay, so this is these are the options. These are what you can mention is something called minus minus FR or minus minus RF or minus minus FF. Okay. So, these are the three options that you have. So, what do they mean is that they actually specify the upstream downstream orientation of these read 1 and read 2 for a paired end data. Okay. So, for example, if you specify minus minus FR, okay, so if you see let us say a paired end alignment, okay, and the mate 1 appears upstream of the reverse complement of the mate 2, right. So, you have mate 2 here, right, and then you see this mate 1 in the upstream region, and the alignment of mate 2 is in the reverse complement. Okay. So, you see this reverse complement. So, this will be a valid alignment, right. So, the default is minus minus FR. We expect the mate 1 will align, right, in forward direction, and you see the reverse complement of the mate 2. The other can happen, other option can happen as well, right. So, mate 2 can appear upstream, right, but in the forward direction, and we see reverse complement of mate 1 downstream, right. That is also a valid option, valid paired end alignment when you have minus minus FR option specified. You have the other options, right. You can see you can think about the opposite one, right, the minus minus RF, which is the reverse complement of mate 1, which is upstream of the forward sequence of mate 2. You can also specify minus minus FF. You expect to see both in the forward direction in some regions, right. So, for Illumina it is mostly minus minus FR, which makes sense. Okay. So, we will not touch this option, but it is good to know that you have these variations. Then you have some of the options that you can use, right. So, one of the things that will happen when you are aligning paired end data in Bowtie2 is that it will first look for something called concordant mapping. Okay. So, we will discuss in a moment what is concordant mapping, and if it cannot find any concordant mapping, it will go for something called discordant mapping. Again, I will explain in a moment. If it cannot find any concordant mapping or discordant mapping, what it will try to do? It will try to find mapping for individual reads. Okay. So, instead of finding mapping for both read 1 and read 2 in paired end data, it will try to find individual mappings of read 1 and read 2. Okay. And it will, if you mention this minus minus no mixed, it will see, it will do, what it will do, it will not try to find these individual mappings, right. So, in some cases it might not be useful to have this individual mapping, because you want to actually get the full paired end mapping, because you have repeat sequences or structural variants that you want to resolve. Okay. You can also mention minus minus no discordant, right. So, if you do not want this discordant mapping, you can simply say I do not want discordant mapping. So, just discard, just stop at concordant mapping. If there is no concordant mapping, just discard those reads and do not do the discordant mapping. In some applications, if you are not interested in looking at this structural variants, right, or some sort of recombination, right, you probably would not do this discordant mapping. So, again you can mention this, and this will speed up the process even further, right. So, it will not try to do the concordant mapping, then discordant mapping, and then do the individual mapping. So, it will just stop at concordant mapping. If it finds concordant mapping, fine, it will report. If it does not find, it will just discard the read. It will not, it will say like no match found or no location found. Okay. So, you will also have other options. These are called minus I options. So, this actually again will specify the fragment length in case of paired end data, right. So, this specifies the minimum fragment length that it requires to call a valid alignment. Okay. So, imagine a situation where you have if you have specified minus I as 60, right. So, you have minus I followed by the number 60, right. So, this is the integer saying that fragment length should be minimum of 60 base pairs. Okay. So, if you have let us say 2 reads, right, paired end, right. So, you have 20 base pair alignment for both in both direction, and you have 20 base pair gap between them, right. So, this will be considered a valid alignment. The default value is 0, right. So, we do not usually impose any minimum fragment length. So, we usually would not change this either. The other one is minus x. Okay. So, sometimes this one could be useful. Okay. So, this is the maximum fragment length for valid paired end alignments, right. So, if you say minus x is 100, right, and if you have so what you mentioning is that we will consider only 100 base pair fragments. Okay. So, we expect the fragments to be smaller than 100 base pair and not bigger than those, right. So, if you let us say you have two 20 base pair alignments on both directions and you have 60 base pair gap. So, that will be a valid alignment, right. So, by default the tool actually takes it as 500, right, which means it will consider fragments of length 500. Okay. Okay, so, I will just mention what is concordant mapping and what is discordant mapping, and then we can go and see how we actually map the reads. Okay. So, as you have seen, right, concordant and discordant mapping, which actually refers to only for paired end sequencing data, these terms. And what is concordant mapping? So, you expect these mappings, right. If you have the read 1 read 2, we will expect this like this kind of location with certain within a certain distance, right. Let us say a fragment length of 500 or 1000, within that you will expect this read 1 read 2 mapping, right. But what will happen, right. So, read 1 read 2 also satisfy this minus FR option, right. So, read 1 is forward direction, read 2 in reverse complement, or read 1 is reverse complement and read 2 in forward direction, right, something like that. Okay. So, what we observe is that okay, read 1 is upstream of read 2 and this is actually what is expected. Okay. So, if we see if the observed is similar to what is expected, right, we call this a concordant mapping. But sometimes what will happen is that this is the expected, but what you observe is that these two reads they map far apart from each other than expected, right. So, you say like they map 10,000 bases apart in the genome, that is very unlikely to happen, right. So, why would you see those two reads map to 10,000 bases apart? Because the fragment length, when you generate the library, we do not have fragment lengths of 10,000 bases, right. Or worse, that they map to two different chromosomes altogether, right, and again this is not likely to happen. Okay. So, this actually refers to discordant mapping, is observed is not equal to expected, right. Now, this is something very interesting in certain situations, right. For example, if there is a structural variant or there is some sort of chromosomal fusion, you are likely to see these kind of discordant mapping, right. So, if you let us say in your data you kind of find similar discordant mapping for many reads, right, you probably can carefully look into that and you can say okay, maybe there is some sort of chromosomal fusion or gene fusion, right, where one read is mapping to one chromosome, the other read is mapping to another chromosome. And if you have multiple observations like that, you can probably go ahead and kind of test that kind of situation. Okay. So, I think that kind of gives you an overview of the options, right. What we will do now, we will just simply look at this run, it is actually quite simple, right. So, what we will do is we will run this Bowtie2. So, back to this terminal where we have created the index files, I can see these index files here, east underscore WG 1, 2, 3, 4 and the reverse ones, reverse indexes, right. So, what we will do, we will use this to actually map these reads against the reference sequence. So, we have to actually call Bowtie2 from that folder, right, because we do not have this system wide installation. So, we are calling this from the folder and I will just call this with minus help, because then it will show all the options that we have just discussed, right, and it will show the usage, right, pattern, how you should run this command. Okay. Here it is, right, and there are a lot of commands, you can of course go through all of them. We have discussed some of the relevant ones. The most important one is here, right, this is Bowtie2 usage, right, how we should run this command. You have Bowtie2 options minus xpt2 idx and then the read names, the input file followed by the output file. Now, in the output file again we will have to mention this the SAM file, right. So, let us do that, right. In the input file you can also see another option which is the -b BAM, right. So, this is some data sets might be in BAM format, right, we have as we have seen, and this can be specified as BAM. Okay. So, in our data it is in FASTQ format, right. So, what we will do is we are calling this Bowtie2 now, okay, minus x the index file base name, right. So, which is east underscore WG right. Now we have to give the input file names, right, the FASTQ files, and remember here we will provide the trimmed files that we created last time, right. When we were doing the hands on, we did some trimming, right. That is the so these are the files that were created after the pre-processing step, right. So these are the files that we will use for mapping, read mapping, right. So the input files will be these trimmed files, right. We have them in that folder. So because we have paired in data, we are using this option -1 iteration 1 read 1 trim dot FASTQ, then followed by -2 iteration 1 read 2 trim dot FASTQ. Okay. And only one thing is remaining is the output file name, which should be in SAM format. Okay. So we have given this -s option and the output file name. I am just mentioning output dot SAM, right. So the alignment will be generated here, right, in this output dot SAM file. Okay. So it will take a very like a few moments to generate, but it is actually very fast, as you will see, right. And we are dealing with a very small data, right. We are only mapping 25,000 reads, 25,000 paired entries. So we have 25,000 pairs, and it is done, right. So if you are dealing with the big data sets, you will see it will take quite a bit of time and much longer than this, but it will still be in hours, right, not in days. Okay. So here are some statistics that we can actually read, right. So it found 25,000 pairs, out of which so it could not concurrently map 2,354 reads. Okay. But it could map about 19,500 reads concurrently exactly one time, right. So there is unique mapping across the reference genome, and there are 3,000 reads for which it found concordant mapping, but more than one time, right. So it means there are multiple mappings, right. They are mapping to repeat sequences in the genome. Then it is looking at these 2,354 that did not align concordantly, right. So it tried discordant mapping. So you can actually stop this using this no discordant option, right. So you can stop this with those options, and then finally it also looked at this which did not align concurrently discordantly. It is looking at independent alignment of the maps, right. Again you can stop this option, right, using this no mixed option that we discussed. Okay. So we will look at some of the variations of this command when we actually discuss the output files, right, and we will see like what are the useful options in these cases. Okay. So here is the reference, right, resources that we have used, and finally to conclude. So Bowtie2 function, we have used this function to map grids, and we have discussed these options in a lot more detail, but as we go along we will see we will actually understand the utility of these options a bit more. And we have also talked about these global local alignment methods, and they have varying speed and accuracy. Again, we can implement them in the subsequent classes. And that is it for today. Thank you.